Thank you for joining the Ones Changing the World, which is India's first Future Tech Meets Sustainability podcast. And today I'm delighted and honored to have with me Jennifer Schwarz, who is a theoretical physicist at the Syracuse University Department of Physics in the Bio-Inspiring Institute. And she also leads the Jen Schwarz Theory Group. So Jennifer, really appreciate you taking time and being on our podcast. Why don't we start with a small brief introduction?
So as you just noted, I am a theoretical physicist. My training actually is more in a phase transition of what we like to call now dead matter. So matter that is not living. In granular systems, how particles go from jammed or stuck to unjammed or flowing. So that's where I began my training. And since then, I've become more and more interested in biological systems and how they work at multiple scales. And so I've been modeling cell nuclei, and there's very interesting dynamics of chromatin in cell nuclei. There's something called correlated chromatin motion, which was observed maybe about 10 years ago. And so building minimal models for how correlated chromatin motion emerges. So, and other scales I've been working on, cell motility, how cells move through microchannels, and also at the tissue scale, which now then brings us to brain organoids, thinking of tissues or organoids from a theoretical physicist perspective as a cellular collective. So a collection of cells, a clump of cells that grows and becomes, if you will, a particular type of organoid or organ, but really organoid, depending on the type of treatment one gives it.
So you spoke about dead matter. Can you explain the difference between non-living matter and living matter?
So, we have a coffee cup here, we have our computers, all solid state. These are systems, if you will, that do not have properties of life in the sense that we would call them, they necessarily can't learn, though here's where we start to talk about AI and sort of the boundaries between what is life, what is living matter versus dead matter. So one would say, a granular system, that's a system of particles that interact, they have some interactions, and then the system can be deformed based on boundary conditions, but there's no sense of a learning capability. There might be actually some memory capability, but there's no sense of learning. There's no sense of sort of coming up with solutions, creating solutions to problems that maybe hadn't existed before. So if we were to define life, what is life? One could say that it's a system, if you will, that with learning and that can learn and approach problems with some sort of creative new solution. Of course, people always talk about, when they talk about life, we need water, we need this, we need that. We certainly, need all of those things. We need fuel for, say, our body. But then the question is, cars also need fuel. So would you consider a car to be living? And I guess I would say, at this point, no, I would not consider a car to be living. But of course, as we start to approach the boundary between machine, so maybe we could call dead matter a machine and living matter a biological type construct, once we start to build either biocomputers via brain organoids, as is happening as we speak, or we also go from the other side and have machines that can learn, we start to blur the boundary between sort of dead and living matter, because sort of maybe each have sort of elements of the other.
Right. Yeah, I think we kind of already blurring the lines. Matter, makes everything. It creates the stars, sun, moon, sand, us, and everything.
We are matter too. We are dead matter
exactly.
to some extent. Yeah.
Yeah, exactly. So, but then, what is that ingredient? What is the bunch of matters that comes together compositionally to create organic life?
Okay, so that's a great question. I've been thinking about it too as the lines become more blurred in the sense of what is the boundary between dead and living matter. And maybe in principle, the boundary isn't a hard boundary, in the sense that certainly for biological systems, there also is in dead matter too, but a a multiscale, a hierarchy of scales. Certainly with dead matter, we start with, well, we could even go, not even start with atoms. We could start with sort of even smaller scale, but we could start with atoms. So we have our coffee cup, we have our tables, we have our grounding material. We have particles, if you will, at the smaller scale, which then interact at that small scale. And then when we think of a collection of them, the emergent properties occur in the sense of being sort of jammed or unjammed or other properties. In the living system, however, there is an active feedback, I think, between sort of the different scales and how, let's say, in terms of biology, so now we get to biology and we say biologists can mutate genes. They can mutate a gene in even an organoid, in a brain organoid, for example, and then one can ask, based on that mutation at that nanometer scale, what happened, what differences, or maybe not so many differences, but what differences can potentially emerge at the centimeter scale? Differences in structure, maybe differences in function, given certainly genetic mutations in brain development. We have lissencephaly, which is development of a smooth brain. Our brains have folds, but with a genetic mutation, one can have a brain that is not as functional, but also does not have as many folds. So from the biology perspective, we change something at the small scale and we have perhaps some outcome that affects some outcome at the bigger scale. Now, there is feedback also, again, the dead matter, in the sense that we could tune interactions at the small scale and maybe have something that a system that maybe wouldn't jam or act a little differently. But the thing is living systems in principle can adapt. And that's where the creativity and whatnot, adaptation, however we want to call it, comes into play. Of course, one could say, but, Jen, I'm going to build a dead system that can adapt too. And so, maybe, that's where, again, the lines become blurred. And so certainly in the biological system, with this interplay of scales and how the different scales interact, certainly that interaction and the feedback between the different scales is much more complex in a biological system, and it's one that we don't understand yet fully, hardly at all. Whereas in the dead matter, we can sort of understand, we change interaction strengths and whatnot between atoms or between this, we can sort of then make predictions as to what happens at the larger scales. And so I would say there's active sort of feedback between the different scales in a living system. In a dead system, you change something small scale and you sort of know the consequences because there's not necessarily complex feedback and active feedback and adaptation and whatnot. So I would say sort of the living systems in terms of adaptability and feedback at actually all scales really kind of gives rise to an organism, if you will, that's much more intricate and complex than something as dead matter.
So Jennifer, you have been working on brain organoids. Would you mind kind of explaining first what are brain organoids and how is it helpful?
Yeah, so brain organoids, one can start out with some pluripotent stem cells. These stem cells are sort of not programmed yet, if you will, to become, well, they will be, they're not yet, if you will, a brain cell or a neuron or a kidney cell or a heart cell. So as they grow, these clump of cells in a petri dish, they're given growth factors. They may be put into a spinning bioreactor. They maybe have some matrigel around them. So certain conditions upon which then they become, if you will, a larger clump of cells with more cells, say of order, 100,000 cells, depending on, how long you wait for them to grow. There's certainly limitations on their sizes, given that cells in the core need nutrients to survive. And as the brain organ becomes bigger, it becomes harder to deliver nutrients to the cells of the core. And as that clump of cells divides, it can take on properties, aspects of, say, for the brain organoid, our brain, aspects such as neuronal diversity, where you can have a whole sort of diverse array of different types of neurons. You can also see layering, as we have a six-fold layering of the cerebral cortex. You can also, if you grow these brain organoids, so this is work done in the lab of Orly Reiner, if you grow brain organoids under confinement, so they're grown in sort of a quasi 2D environment, you can have these organoids actually develop some indications of fold-like structures, just like we have folds in our own brain. But this is under sort of a certain set of conditions. So there are some structural similarities, and then you also come to functional similarities in the sense of as these brain organoids become older, in the order of sort of months old, actually these neurons that emerge can actually fire electrical signals. And so then once you start having a working neuronal network in a brain organoid, of course, then that begs the question of, oh, let's build, brain organoid computers.
Wow, how cool and fascinating is this that you take a cell, you put it in a petri dish, create a pluripotent stem cell, and then could put it in a petri dish and, give it, the things to kind of
help
mature
it grow. Right.
and eventually it becomes these tiny clumps of brain organoids, which resembles the patient's own brain, and you can do like anything. You take the cell and you can create a kidney, you could create a different organoid also besides. So how fascinating is that? Could you elaborate a little bit more on this? When you create this brain organoid, you're saying that structurally, work as well as functionally, it can resemble the patient's the brains, and how whatever it's doing, the neuronal fires, if it's doing that, how much can we learn of the patient? And if we have kind of this, if you're modeling this is a model of the patient's brain, what can we learn from it, and what are the ways that we can create applications which could help in the healthcare space?
So certainly, yes, the precision medicine is almost, is here. So indeed, one can take a patient's blood, create the pluripotent stem cells, and then grow, if you will, a brain organoid. Say if the patient has a glioblastoma, and then one can ask, okay, now given this brain organoid, or this brain tissue, if you will, it's in the petri dish, presumably one can test out different treatments to see whether or not at least this particular tissue in the petri dish will be responsive or not to particular types of treatment. So one can have tailored, sort of individualized treatments depending on what happens in these experiments. Now, there is an issue, though, of course, with glioblastoma. Depending on the progression of the disease, it's on a very fast timescale, maybe on the order of months. And at least as of now, there are issues with timescales in terms of growing brain organoids from a patient's stem cells, in the sense of maybe it's sort of more like a year-long timescale. And so these things takes time. Of course, that timescale will probably shrink as technology improves. But here is where I would like to step in the sense of, as a theoretical physicist, and computational physicist, I build computational models. And so here is where, maybe, if I sort of know enough about the patient, or maybe they sort of are able to, if you will, get some pluripotent stem cells, then maybe I can, as a simulator, make predictions ultimately about, so simulate the growth of the brain organoid based on a person's, if you will, chromatin organization and whatnot, and then simulate in the computer on a much shorter time scale, and try to make predictions about potential treatments. Now it's still a little far away in terms of simulating in the computer the growth of the brain organoids and the structure and the function, but that's what I'm working towards, myself and other collaborators. And another thing would be to help understand what are the emergent properties of learning, and to do this through looking at the emergence of a neuronal network in a brain organoid. Now, there's all sorts of questions about brain development, and people for a number of years now have mentioned sort of an adult human brain the connectome has a small world network architecture. So one could ask, that's a beautiful finding, and maybe, that's sort of being tweaked and whatnot, but how does that particular neuronal network architecture emerge? Now, it might be in the brain organoid that network architecture is a little bit different. That's what sort of is now work being currently explored. But the question would be how does that small world network emerge? So in addition to sort of helping from a medicine point of view, I'd also like to understand sort of how does a brain build itself? So what are the mechanisms behind brain development? And so I can sort of certainly talk to experimentalists, and I can also, from physics, I can try to build models, minimal multiscale models, upon which ultimately one can explore how these neuronal networks emerge in these brain organoids. And then based on their architecture, what functionality are they capable of? And how does learning, sort of, as I like to say now, the emergent properties of learning in living and non-living systems, because one can also study the emergent properties of learning, as people are doing in AI, or even in physical networks, like spring networks. And so then one asks, what is the difference between learning in a spring network or a network of resistors versus a network of neurons? And so with the brain organoid, we have this beautiful sort of capability, because we don't need to involve people, we don't need to involve animals, it's cells. And so sort of many, many experiments can be done. And that can also provide then sort of a great testing ground for computational modeling.
With the computational model that you're talking about at this point in time, could you kind of elaborate on that? Because that sounds really fascinating, because when you're kind of taking a cell and creating a pluripotent stem cell, when you take it in a petri dish, this takes time to mature. But then if you have a computational model, once you kind of understand the architecture, that could really be a game changer. Would you like to elaborate on that?
Yes. So maybe I'll be just a little bit specific for the moment, and then I'll be a little more broad. So here is a particular question in terms of brain development. So a question could be, which many people have been asking through the ages, how is a human brain different from a chimpanzee brain? Right? So we have different species. Let's maybe we have mammalian species. So we have sort of a mouse brain whose volume is of order, something a tenth or so, or 100th of a centimeter cubed. We have a human brain. It's of order, people say 1,500 cubic centimeters. And then we have a chimpanzee, which is about a third of that volume. And so, one could ask, what sort of, we obviously are different species. What's separating us? And so people certainly hone in on, given that our genome is very similar to a chimpanzee, meaning we're sort of a 1% difference in our genome, how is it then that we have sort of a 3 times sort of a larger brain than a chimpanzee? And is that larger brain enabling us to sort of do more things, if you will, to create more, to invent more? Certainly chimpanzees also have been found to invent things as well, but we do it, I guess, on we could say, a larger scale. And so what are the differences between a chimp brain and structurally and a human brain? And so that question, of course, has been now reframed in the context of brain organoids. How is a chimp? So if you take pluripotent stem cells derived from a chimp versus pluripotent stem cells taken from a human, you take those cells, you have their respective petri dishes, and you grow brain organoids in the 2 respective petri dishes. Madeline Lancaster's and in her lab has found that indeed that the human brain organoid, the area, if you look at the surface area or the projected area, is larger, say even around day 10, than a chimp brain organoid or a chimp-derived brain organoid. So she has discovered that a potential reason for this difference is that there is a transcription factor, which is called ZEB2, that is upregulated during the time at which the cells in these brain organoids take on a very different shape. And that transition, when the cells take on a very different shape, happens later in the human brain organoid, meaning at around day 10, as opposed to day 5 in the chimp brain organoid. So the point is there is this finding of upregulation of this particular transcription factor. So that's at the gene scale. And then that translates to something change in the cell shape scale. And then that cell shape change, where the cells go from sort of more globular to being longer and skinnier, then happens at different times in the chimp and the human. And so she's kind of traced that cell shape change transition when it happens as a key indicator for the differences in size. So now one asks the question, and here's where I step in. Okay, so if there's sort of this difference in gene expression, the timing of it, what is happening? And so really, can we link the gene sort of scale to the I guess more of order 200 micron scale or whatnot in terms of the brain organoids, because they're kind of younger brain organoids, meaning up to day 10 or so. And so what we've proposed as a hypothesis is that as the cells divide in the brain organoid, what a potential hypothesis, the difference between this delayed and onset of the cell shape transition, is that there can be maybe a chromatin reorganization due to the cells dividing in a confined space. As the cells divide, they become more crowded. And as they become more crowded, the cell shapes change a little bit. And then therefore, the nuclear shapes change. As the nuclear shapes change, that can have trigger some rearrangement in the chromatin. Chromatin is sort of the DNA plus histone, sort of how the DNA gets all sort of put into the cell nucleus. And so if you have a physical mechanism of sort of nuclei changing shape, that can trigger chromatin reorganization, which then maybe can trigger the ZEB2 expression. So it's a hypothesis, really, it's a multiscale hypothesis that asks the question, sort of what's really happening at the chromatin scale between the chimp and the human? Can we see changes in chromatin organization earlier in the chimp that then triggers the cell shape change as compared to the human? And what I am trying to do is then build models where we have chromatin, that is balled up into a cell nucleus, a deformable cell nucleus, that then is embedded into a cell, a deformable cell, that then is now attached or interacting with a collection of cells. And then those collection of cells divide, and as they divide, we can keep track of the cell shape, we can keep track of the nuclear shape, and then we can keep track of the chromatin and how it reorganizes. And then the last final step will be relating chromatin, sort of that sort of slightly hundreds of nanometer scale, if you will, to the gene scale, which would be of the order of the nanometer scale. And so with these computational models, we can certainly test this hypothesis. Do we see chromatin organization happen earlier in a way earlier in the chimp as compared to the human, and in particular the area of the chromatin that's sort of related to the ZEB2 expression ultimately. So that's what I can test in the computational model. Of course, Madeline Lancaster in her lab can test this hypothesis experimentally, because there's a technique known as Hi-C, which actually can give one in a real world experiment, what does the chromatin organization look like in these cells as the brain organoid develops over time, chimp versus human.
I'm sure there are other teams also who are working on brain organoids. Could you talk about some of your peers who are at the forefront of this, and what are the problems they are solving? How do you think that's going to impact the future of healthcare?
So in the labs of Orly Reiner and Amnon Baksim, who are collaborators of mine, wonderful collaborators, brilliant people. So they have performed a genetic mutation that's related to lissencephaly, and explored the effects of that genetic mutation in brain organoids. And they have found that actually the brain organoids, there are differences between the wild type or the non-mutated versus the mutated. There are differences in mechanical properties of the brain organoids. And that in itself, so being a physicist and mechanics, forces, energy, that sort of, those notions interest me. And so I am working with them to understand, how those mechanical differences in these organoids at day 10 and maybe tens of days later on as well, in those organoids, what can in principle those mechanical differences translate to in terms of functional differences? I mean, that certainly ultimately structure is beautiful. We want to understand structure, but then we want to link that structure to function, as the biologists always say. So that sort of would be the next step. Ken Kosick, a brilliant neuroscientist at UCSB, and in his lab, they've been working on the electrical signaling of brain organoids, looking at the neuronal network, looking at the architecture, as I mentioned, trying to understand sort of again, I don't want to steal anyone's thunder, but understand the robustness of these neuronal networks. I and Ahmad Borzou are working on trying to understand the neuronal avalanches in these brain organoids. So when you can ask what's the size of that avalanche, the total integrated signal, and you can actually plot the distribution of those avalanche sizes. And people have done this for rat brains, slices of rat brains. People have done this for maybe since 2010, maybe was the first, 2003 was maybe the first paper where they looked at a rat brain, and they found actually a broad distribution of avalanche sizes in rat brain slices when you stimulate them. And so one can ask, do we find the same type of distribution in a brain organoid? Again, trying to sort of explore similarities and differences between a brain organoid and a brain.
My last question, what next to you? Because we're running out of time, maybe just a couple of minutes there. What comes next to you? And you're also working on organoid intelligence, biocomputer, right? Maybe you could just address that.
My collaborators and I have now an algorithm that we call frequency propagation. So this is with Videsh Rao Anisetti, Anna Kandala, and Benjamin Chillier. We have a new algorithm called frequency propagation, which is rooted in a physical system, which is different, related, but different to, say, backpropagation, which is what the machine learning people use to train a neural net to be able to learn how to recognize or distinguish between a cat versus a dog. So as we work on understanding the emergent properties of learning in a physical system or a dead system, and really understanding fundamentally what is key here. This is where, as a physicist, we want to understand the basic underlying mechanisms. Physicists have worked toward understanding what's the underlying mechanism for the basis of mass and whatnot. Here, we want to learn what are the underlying basic mechanisms for learning at its core. So, because if we can understand those such mechanisms at their core, then we have the capability to leap above and beyond, what the current understanding. So just as we'd like to learn about learning in dead systems, we also like to apply, say, this frequency propagation algorithm that we introduced recently to living systems, to apply these types of algorithms to a brain organoid, the neuronal network in a brain organoid, to determine how a brain organoid learns. So it's not just the fact of demonstrating that it learns, that is great, but what is actually going on? What is underneath the hood? And looking for universal, if you will, mechanisms across living and dead systems. So again, blurring the boundaries between living and non-living matter. So that sort of is where I'm next headed in terms of organoid intelligence, if you will, or organoid learning. And that, of course, also involves how does the architecture affect the learning? And so that's where the architecture of the neural network, certainly that's where we come back to structure. And so again, going toward this sort of gene to tissue structure puzzle. So thinking about that as well in terms of how we relate this multiscale setup of really, if you will, computers within computers, that's sort of a feedback between them and adaptability, that's sort of a living system for you. And so how the structure then, building these multiscale computational models to understand and be able to predict ultimately the structure of a brain organoid we'd like to then couple that structure to ultimately how does the neuronal network evolve, and then based on the architecture of the neuronal network, what is the functionality of the brain organoid in terms of its capability to learn. So that's what's up next. So stay tuned, and thank you very much for talking with me. And I guess I'd like to acknowledge also in this structure approach in terms of linking ultimately genes to tissue, though right now it's more chromatin to tissue. I'd like to also acknowledge great collaborator Tao Zhang of Shanghai Jiao Tong University in helping build the computational models with me, as well as Sartik Gupta and Ali Pattison and Bannigan. So thank you.
Lovely. Jennifer, really appreciate you taking time and being part of the podcast. To my listeners, if you like what you see in here, then please press the subscribe button. Until next time, see you guys. Bye-bye. Thank you.